And welcome to my YouTube channel. So here today we are going to be looking at higher fungi. Also, we are looking at the other one, which is Basidiomycetes. So what is Basidiomycetes? So Basidiomycetes are mostly macroscopic, meaning to say they are huge. Macroscopic, they are huge. For example, we can have something like this. So this is basically, most of them are mushrooms, but not always. So they are mostly mushrooms. They are huge like that. So which leads us to our other characteristics. So they are sometimes called basidiocarp, and also they are able to produce a structure which we call basidium. So basidium means one. Basidia is when there are a lot. So as you can see, you can point here, but there are a lot of them. So these structures are the ones that are able to bear spores. So the basidium are the ones that bear spores. So the one basidium will be able to bear as much as 4 spores. So 4 spores. So the predominant thing here is sexual reproduction. So that is what we are going to be discussing. So Basidiomycetes is made up of a network of mycelium. Remember, mycelium is just a network of what we refer to as hyphae. So when there are a lot of hyphae here, so as you can see, these are hyphae. Then the one that bears fruits, the fruit bearing part, is what we call the basidiocarp. So these, when you extract it, you are able to get the basidia. So basidia is a lot of these. So each basidia, remember, it is able to produce 4 spores. So now, with that said, let's move on to the sexual reproduction. Okay, so let's begin here. So this is our basidium. So this basidium is able to produce 4 spores, as you can see. So these spores are what we are going to be calling Basidiospores. Other books will just use the word spores. So same here, I just repeated it, but they are Basidiospores. So these Basidiospores will now be able to grow, or rather, so this one is germination. So germination is where they will grow and produce hyphae. So as you can see here, this is our hyphae, like that. So remember, hyphae is formed from the spores. When they grow, they produce hyphae. So now, when when these hyphae, they are able to germinate, but the best term that I would prefer we use is differentiate. So here you can use germinate, but here you can use differentiate. So the hyphae will be able to differentiate. As you can see, at first they were just the same. So let me even do this. So at first they were just the same, like that. So these 2 hyphae were the same. Then after that, they will be able to undergo differentiation. Differentiation is where now they will be able to become positive and the negative mating type. So under Basidiomycetes, we use positive and negative. That is the names of the males and the females. Then the main difference is the Basidiospores. Then these will be able to form positive and negative mating types. Then afterwards, they will be able to undergo fusion. So they will come together, fuse like that, and they will be able to form what we call plasmogamy. So plasmogamy is simply what we refer to as the fusion of the cytoplasm. So the cytoplasm fuses. As you can see, there are 2 different nuclei. So during plasmogamy, the nuclei are different. So remember, so plasmogamy, by definition, I'll repeat, we are saying that plasmogamy is simply the fusion of cytoplasm. So be careful, don't say the fusion of the nucleus. Then after that, when conditions are favorable, they will be able to form the fruit part, which we are going to call the basidiocarp. So this basidiocarp will be formed when the conditions are favorable. So you can say that, so after plasmogamy, they will be able to become basidiocarps if conditions are favorable. So if conditions are favorable like that, they will be able to become what we call the fruit part, which is basidiocarp. Then the basidiocarp, as you can see, will be able to become what we call the zygote through the process of karyogamy. So karyogamy is the fusion of the nucleus. So don't forget, karyogamy is the fusion of the nucleus. Then this process, there will be meiosis. So mainly you can even say after the zygote has been formed, then this one is what we call so this is an extract from so this is an extract. So as you can see, this is basidium. Even this is basidium. Basidium will be able to produce 4 spores. So it is more like it is just an extract from this to take it there. So under this will be able to undergo meiosis to produce spores. So these spores are genetically different, and these spores that are produced are Basidiospores. So sometimes, just as I said, they will just call them spores. And these spores are haploid spores, meaning to say it contains half the number of chromosomes. So after that, the cycle will continue. Again, from the beginning, we have the spores. So we were on spores. So these spores will be able to germinate. So remember, these spores will be able to germinate. When they germinate, they will be able to produce hyphae. These hyphae will now be able to undergo not germination but differentiation. So you can replace this word with differentiation. So they will be able to undergo differentiation, which is they become specialized into male and the female mating type. So remember, for Basidiomycetes we use positive and negatives. Then from there we are going to have plasmogamy, in which these two will fuse to share the cytoplasm. So plasmogamy is the fusion of cytoplasm. So when the cytoplasm fuses, that is what we call plasmogamy. So fusion of the cytoplasm. Then after that, when conditions are favorable, they will be able to form what we call basidia. So basidia, or should I say basidiocarp, which is the fruit part. Then the basidiocarp, you are now going to have what we are going to call the basidium. The basidium, which is able to produce 4 spores, as you have seen here. So before that, we have the karyogamy. Karyogamy is where now they will be able to form the zygote by the fusion of the nucleus. Those 2 nuclei that were different, they will be able to fuse to produce what we call the zygote. Then from there we are going to have the Basidiomycetes releases the Basidiospores, and the cycle continues like that. So this is another diagram. So we can begin from the basidium or the Basidiospore. So this Basidiospore will be able to undergo karyogamy to form diploid nucleus, meaning to say, from here, as you can see, where can we begin? Okay, let's begin from Basidiospores. Basidiospores will be able to germinate. When they germinate, 2 mating types, positive and the negative. So positive and the negative. Then they will be able to undergo plasmogamy, which is the fusion between positive and negative mating types, resulting in the in the formation of a dikaryotic mycelium. Dikaryotic meaning to say it has got two nuclei like that, two different nuclei. Then after that, under the right environmental conditions, a basidiocarp forms. So gills of the basidiocarp contain cells called basidia. So this is what we call basidia. So basidia are a lot, basidium is one. Then the fruit part, the whole thing is called basidiocarp. Then we have basidia there. So they will be able to fuse under the process of karyogamy, in which there is the diploid nucleus. Here we have, remember, here we have a dikaryotic, meaning to say there are two nuclei. Now diploid nucleus means the two nuclei are fused together to form what we call basidium with four nuclei under the process of meiosis. Then the cell divides, producing four Basidiospores like that, and the cycle continues like that. So that's that, dispersal and germination. That's that. Thank you very much for watching. Hope you would like, subscribe, and share for more. See you in the next tutorial.